Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR Net JRF Life Science question paper shift 2. These are part C questions. This was conducted in the year July 2024. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of CSIR been answered, you can go to my channel Omni Bioentrance. In the channel playlist, C for CSIR Net JRF, there you will find these. First question. The figure summarizes the observation following a cross between two haploid strains of Neurospora crassa having alleles A, small a respectively. The following statements were made. Now this is capital A and this is small a. Now if you see the first one, first assay, you will see that it is 2:2:2:2:2, showing that they are recombinants. Okay, because 2:2:2:2 combination you will get in second generation segregation, whereas 4:4 or 1:1 you will get it in first division segregation. So this is a second division segregation. Then this one, it is 2:4:2. Again a recombinant, again a second division segregation. Here also 2:4:2, again a second division segregation. This one 2:2:2:2. Again a recombinant. But here you see this is 4:4. This one also 4:4. So they are non-recombinants. They are first division segregation. Now see the statements. In first, the segregation of allele occurred in the anaphase 2. Yes, in first assay they occurred in anaphase 2. That is why it is showing second division segregation ratio. So first is correct. Crossing over between the centromere and the gene occurred in 20% of the myocytes. Okay. Now how will you calculate that? You calculate the number of recombinants by total number of offspring into 100. That is 16 plus 14 plus 18 plus 12. That will give you 60. 60 divided by 300 into 100 will give you 20%. So that is also correct. Crossing over between centromere and the gene occurred in 20% of the myocytes is correct. With reference to the non-recombinant parental chromosomes, there are 6 different ways by which they can orient themselves at the equatorial plate. Actually, for orientation of the chromosome at the equatorial plate, there is a formula, that is 2 raised to n, where n is the number of chromosomes per set. So it is not 6, it will be more than 6. So C is not the right answer. The gene is 20 centimorgan away from the centromere. That is also correct, because we got here that crossing over between myocytes and the gene occurred in 20% of the myocytes. So D is also correct, but it is not there in the options. So A and B only they have given. So 1 is the right answer. Then next one. Match the major cell cycle regulatory proteins in column X and their typical function in column Y. V1. V1, it is phosphorylates inhibitory sites in CDK. A2. P27 suppresses G1S CDK and SCDK activities in G1 phase. B4. A2, B4, A2, B4, A2, B4, 1. So 1 must be your answer. A2, B4. C1. P21 suppresses G1S CDK and SCDK activities after DNA damage. Yes. CDH1 phosphorylates, activates APCC in the late mitosis and early G1 phase. C3. D3. So A3, B4, C1, D3. So answer is option 1. Then next one. Protein phosphatase 2A PP2A is a critical regulator of CDK1 substrates during the cell cycle. Okay. B55 subunit of PP2A influences the substrate selectivity, localization and regulation of the enzyme. Given below are few statements about PP2A and its regulation during the M phase of the cell cycle. So what you should know here is that MCDK1, it activates what is called as the great wall kinase, which inhibits the B55 subunit of the PP2A. Okay, so PP2A, P25 is inhibited by the great wall kinase. Now, when the anaphase is initiated, CDK1 is inactivated. Now, when CDK1 is inactivated, great wall kinase cannot act. So, this relieves the inhibition of PP2A B55, which are phosphatases. They have already told in the question, and they will dephosphorylate the CDK1 substrate. Okay, so these 2 points you have to remember. Now read the options. PP2A B55 activity is high during interphase but inhibited during early mitosis when CDK activity rises. Yes, because of the presence of MCDK1, which activates the great wall kinase. So first one is correct. MCDK1 turns off PP2A B55 via the phosphorylation of an intermediary protein kinase called great wall. Yes, B is also correct. Just now I told you. MCDK1 turns on. No, it turns off. So C is not correct. When anaphase is initiated and MCDK1 activity declines, PP2A B55 promotes dephosphorylation of CDK1 substrates. That is also correct. I told you because the great wall kinase activity reduces when MCDK1 activity declines, and PP2A B55 will be free and it will start dephosphorylation of CDK1 substrates because it is a phosphatase. So A, B, D are correct. Answer is option 1. Then next one. The following statements refer to mechanisms that may confer resistance to antibiotics in bacteria. Enzymes that can break down the antibiotic. Yes, they will confer resistance to antibiotics in bacteria. Efflux system to pump out the antibiotics. Yes, that's also correct. CRISPR mediated defense against the antibiotic. CRISPR Cas9 can be used to reduce antibiotic resistance in bacteria and not mediate defense against the antibiotic. Okay, so C is not correct. Antitoxin that can sequester antibiotic, that is also incorrect. Cell wall modification, E is correct. So A, B, E is the answer. Then following statements are made about vaccine. Covaxin is killed cell vaccine and Corbevax is a subunit vaccine. That's correct. Oral polio vaccine is given as live attenuated vaccine to adult and as killed vaccine to children. No, to children also live attenuated is given. So B is incorrect. Third generation vaccines against smallpox are based on attenuated vaccinia virus. That is correct. MMR vaccine is given to children to protect them against diphtheria. Not diphtheria but measles, mumps and rubella. So D is also incorrect. So the correct answer is A and C only. Then next one. Given below are few statements on related to inheritance biology. Quantitative traits are characterized by discontinuous variation in the phenotype. It is continuous variations because you can take the example of a wide range of heights in humans across the world. They show a continuous variation, not discontinuous. So A is not correct. Polygenic traits never show normal distribution of phenotypic variability. They show normal distribution. So B is also incorrect. So what is left out? C and D. C and D is there in the option. So 4 is the answer. Wherever A and B comes, they are wrong. Association mapping captures wider genetic diversity than biparental linkage mapping. That's correct. Bulk segregant analysis can be used for mapping of monogenic qualitative traits. That is also correct. Then next one, deletion mapping. Mutants of bacteriophages that carry deletions can be used to rapidly locate mutation sites of newly obtained mutants. The mapping is based on whether wild-type recombinants can be recovered when the deletion mutant and the novel mutant are brought together. For independent deletions 1 to 4, that is this, of a region were used to map 4 novel mutations A, B, C, D. The deletions starting from a fixed site are shown below. The lines denote the region of deletion. The results of mapping are summarized in the table, where plus denotes the recovery of wild-type recombinants and minus inability to do so. Further, it was observed that out of the 4 novel mutants, no relevant revertant was observed for mutant A. Mutant B and C do not complement each other. Okay. Now we will see here. First we will discuss the order of the genes. So I have written the table here. This is the table that they have given A, B, C, D. Okay, and this is the deletion. These lines are the deletions. Deletion 1, 2, 3 and 4 I have drawn here for easy understanding. So in the first one, A is absent, but B, C, D are present. So A is deleted. So A here. A, it will be A there in the first one. In the second one, A and D are deleted. So up till here, it should be A. So this region must be of D. Okay, so A and D. In the third one, A, B and D are deleted. You know the position of A and D now. So whatever remaining will be that of B. In the fourth one, A, B, C and D are deleted. All are deleted. So up till here it is A, till here it is D, till here it is B. So this remaining will be C. So what is the order of the mutation? It is A, D, B and C. So option B, if you see, mutations can be ordered as A, D, B, C. That's correct. Mutation A lies within the region of deletion 1. That is also correct. We just now saw deletion 1. This is deletion 1. So mutation A is present there. Here 2, 3, 4, everywhere it is A. So deletion 1 is A. So that's correct. Mutations could be a mutation A could be a deletion. That's also correct. So A, B, C are correct. Then mutants B, C are located on 2 independent cistrons. It is not on independent cistrons but on different regions. So D is not correct. So answer is A, B and C. Then next one. A cross was made between wild type female Drosophila melanogaster and mutant males which are yellow bodied cross veinless. So males are mutant, females are wild type. The 2 genes are present on the X chromosome. So yellow body and cross veinless, they are present on the X chromosome. The F1 progeny was submated and observation of F2 progeny is tabulated below. So this is F2 progeny. Okay, with regard to the above analysis, which one of the following statement is correct? The genetic distance between the two genes is 7.5 centimorgan. Now, which are the recombinants here? That is 6 plus 9. They are recombinants. By total progeny is 100. 45 plus 40 plus 6 plus 9 is 100. So, 6 plus 9 divided by 100 into 100 will be 0.15 centimorgan. Sorry, it will be 15 centimorgan. Okay. 6 plus 9 is equal to 15 divided by 100 into 100 will be 15 centimorgan. So the genetic distance between the two genes is 7.5 centimorgan is not correct. If the mapping was done with the third marker which lies between Y and C, that is between the yellow body and cross veinless, there was a third marker there, that is a 3-point cross was conducted. The genetic distance is likely to increase but never decrease. That is correct. If it was 3-point cross, then genetic distances will increase but not decrease. If a larger progeny size was analyzed, more double crossovers will be identified, of course, leading to decrease in the genetic distance, not decrease in the genetic distance and increase in the genetic distance. So 3 is also not correct. If a reciprocal parental cross was carried out, no recombinance would be observed in the F2 progeny as there is no crossing over in Drosophila melanogaster males. Reciprocal cross means mutant female into wild type male. There will be recombinance because females can undergo recombination. So 4 is not correct. Then next one. In a plant species, the following pathway contributed to seed color. The wild type phenotype of the seed color is red. See, it is red over here. Now, A gene is contributing yellow pigment in the endoderm and B gene is contributing red pigment deposited on the outer layer. Okay, that is the pathway. So, both these genes are necessary for seed color to be red. Then a recessive mutation in gene A lead to white color pigment. Now this will be white. A recessive mutation in gene B lead to transparent outer layer. Now if there is mutation in B, this region, this marked red region will be transparent and the color of the seed is based on the color of the endosperm. So if A is not mutated but B is mutated means it will be yellow seed color, not red. Okay. Now if A is mutated, it will be white and B is not mutated means it will be red seed coat color. Like that. The two genes are present on two different chromosomes. Okay. Often yellow or white colored seed has red spots. Yellow means it is not mutated, wild type A is present, or white color means A is mutated, so endosperm will be white in color. And they will have, instead of the red coat color being continuous, they will have red spots. Now what does these spots indicate? It shows that there was a transposon present in the B gene, which might get excised from that region in the B gene and it will move to it will jump to some other place. So because of that, the B gene, there will be red spots coming over there instead of a continuous red coloration. Okay. So remember that point. Now read the statements. The probability of getting red colored seeds from the dihybrid cross involving two heterozygous mutants is 9:16. Now heterozygous means it is capital A small a, capital B small b. Now for red color to occur, both these genes have to be present in the dominant state. That is A should be dominant, B should be dominant. Only then the seed color will be red. that they have already mentioned here showing this pathway. So it is not a Mendelian cross. So you will not get 9:16 ratio. The mutation in gene B could have been caused by a transposable element. That is correct. That is why when the transposon got excised, you got red spots, as in this statement. So B is correct. A is not correct. A plant producing red seeds would breed true for the seed color. Need not. If it is a heterozygous, you will get different variations of the seed color. But if it is homozygous, capital A, small capital B, then you will get true seed color. So a plant producing this will also produce red seeds, heterozygous condition, homozygous condition also will produce red seeds. So a plant producing red seeds need not breed true for the seed color. So only B is the correct statement. Answer is B only.